In this lecture, we will be going through the pathway of cellular respiration, kind of working through this under the impression that you already have an understanding of glycolysis and the citric acid cycle. Let's start with a small overview of what cellular respiration is. First, it is a pathway that is going to harvest energy in the form of ATP, which is adenosine triphosphate, from fuel, which is going to be glucose or sugar. And kind of a good way to visualize this is the illustration and picture below. So the gentleman eating the burger, that is where we're getting that fuel or glucose, and we're turning that into ATP or energy. The importance of cellular respiration overall is to carry out all the processes of the cell using that energy that we extract from that fuel that we take into our bodies. So when you think of what a cell does, I want you to think of maybe your heart right now. What's it doing? If you're alive, it's beating. Those cells that make up your heart muscle need energy in order to perform those processes in order for your heart to beat. Same for any organ and any part of your body that's made up of cells. Let's briefly summarize the steps leading up to oxidative phosphorylation, which you have likely already covered prior to this lecture. So the first step is glycolysis, which is going to happen in the cytosol of the cell, which is kind of the juice of the cell, if you will, where glycolysis literally translates into sugar splitting. It's where we take a glucose molecule and split it in half and create two pyruvate molecules. From there, in that process, we're also going to create two NADH molecules, which are going to be electron carriers, and this is going to be important when we get to oxidative phosphorylation. So I've highlighted it in red, so that is a product that we just want to remember that it has been created and we're going to use it later. From there, we have also created two ATP molecules. This is a very small fraction of the amount of ATP that we'll create in that final third step of cellular respiration. The next step I've labeled as step 1.5 because it's kind of a little half step. It is pyruvate oxidation, where we have now moved into the mitochondrial matrix, and this is where this is happening. So this is where we take pyruvate and that we've created from glycolysis, and we will convert it into acetyl-CoA, which is a coenzyme. This is going to be important for the next step. So moving into step 2, which is the citric acid cycle, also used to be known as the Krebs cycle, which is happening in the mitochondrial matrix, just like the pyruvate oxidation half step. So what happens is we take that first molecule of acetyl-CoA and it will bind to oxaloacetate in order to make citrate, which is where the cycle gets its name, which eventually will create another oxaloacetate, which will then bind to the second acetyl-CoA as it comes into the cycle. So in total, the citric acid cycle is going to spin twice, and in that two cycles, it's going to create 2 ATP, 6 NADH, and 2 FADH2s. So the 6 NADH and 2 FADH2s are going to, just like with glycolysis, those electron carriers are going to be used in that third and final step in cellular respiration. Another important thing to make note of is that through the citric acid cycle, we are creating CO2, which is a byproduct. It's not the end goal, but it's a byproduct, which is why we breathe carbon dioxide out as a waste, essentially. It's kind of neat. So from here, we enter into that final step, step 3, oxidative phosphorylation. which I want to point a few things out before we look at it a little bit more closely. One, oxidative phosphorylation happens in two little sub-steps. So we have electron transport chain, which you can see on the left, and chemiosmosis, which happens, is kind of illustrated on the right. And what we see is that these molecules that we want to focus on, which are proteins and an enzyme, they're embedded in that inner mitochondrial membrane, which I've labeled in green. This is very important to make note of. Some of the products are being pushed out into the intermembrane space. So this is the space between the two membranes of the mitochondrion. So kind of keep that in mind. And then we have that more yellowy space, which is the matrix of the mitochondria. So kind of keep that in mind as we work our way through. That oranger area is that intermembrane space, but we're still within the mitochondrion. And the yellow space is the matrix of the mitochondrion. Starting with electron transport chain of oxidative phosphorylation, it is a chain of proteins labeled 1 through 4 that are progressively more electronegative as you progress through the chain itself. So 2 is more electronegative than 1, but 3 is more electronegative than 2, meaning that these molecules are very greedy or hungry for electrons. They want them. So what's going to happen is that, remember that we have in total 8 NADH and 2 FADH2 electron carriers that are carrying electrons. And they're going to come up to this electron transport chain and enter into the chain at various molecules. So it's important to follow the NADH first. So we can see on that first protein that the NADH is going to come in at that first protein, where that protein will pop off the electron that the electron carrier is carrying, and then move the electron from that protein to the next, and then to the next, and then to the next, until we get to the end of the chain in that last protein, where the electrons that have popped off from the NADH, the electron carrier, it's going to find an oxygen molecule, which this is very important because oxygen is the most electronegative and will very greedily accept those electrons. And when it does, it turns into H2O. So water is going to be a byproduct of this electron transport chain. That's kind of important, but gentle reminder, just like CO2, it's a byproduct and not the end goal. So with that, what else is happening? Well, the FADH2, that other electron transport, or the other electron carrier, pardon my misspeaking, is going to take that second molecule as that starting point. So it's not going in to the first protein, it's going in at the second protein in the chain. This is important. I'd make a little star next to this because this will come into play later. So the electrons that are pulled off of those carriers and are moved through the chain, because remember, each protein in this chain is more electronegative than the last. So it is pulling the electrons along the chain. What this does is it creates energy. And then that energy is used as a pump to pump hydrogen ions across the inner mitochondrial membrane from the matrix to the intermembranous space. And this is super duper important because it's creating that hydrogen ion gradient. But why do we care about that? Why do we want a hydrogen ion gradient? As a little reminder of what the big picture of oxidative phosphorylation is, this is that wider view. So we have just covered the electron transport chain, where we can see that hydrogen ion gradient being formed, and those hydrogen ions collecting in the intermembranous space. This is very important because you can see with that little red dotted line where they're going to go to next, which is going to be chemiosmosis, which, let's talk about this term for a second. So we've seen the term osmosis already, which we know is the movement of water. But let's look at the word osmos. So that is Greek for push. But instead of thinking of an active push, I want you to think of kind of more of a passive fall that might happen with a push. Because what is going to happen within this process is the gradient that we've created of hydrogen ions is going to drive this movement. And it is a passive movement, meaning there is no energy required to cause it. So the molecule that we're looking at in its totality is ATP synthase. And what ATP synthase does is it synthesizes ADP into ATP. So in and of itself, ATP synthase is an ion channel because it allows for those hydrogen ions to pass through it, as well as it's an enzyme, and we'll see why. So here we have a ATP synthase molecule, but in substantially more detail than that last image. There are a couple parts of the ATP synthase that we do want to go over, and we will start with that first part. So the rotor, which is that top portion, contains binding sites for hydrogen ions. And those binding sites will change shape, which, if you remember, when we've talked about enzymes, that is an induced fit. And they change shape when the substrate is in the place of that binding site, right? So what happens when the shape changes when the substrate is in the binding site is that the rotor will actually spin, which is super cool. When the rotor spins, it causes the next part, the internal rod, to also spin. This is important because the internal rod is attached to that third part, the catalytic knob, which does not spin. And the reason that it does not spin is that the internal rod, which is in the catalytic knob, is going to create energy. And the reason we want to create a little bit of energy here is so that within the catalytic knob, we can create ATP by phosphorylating ADP. So we're adding a phosphate to ADP to create ATP, which is kind of our big moment here. But why does everything kind of like stay in place and you know what's kind of happening? A lesser player that is our fourth part in our ATP synthase is the stator. It acts as a channel for the hydrogen ions to come into the rotor as well as to exit the rotor. So it is that ion channel portion of ATP synthase, which it will also act as holding the catalytic knob in place as well. So in the end, oxidative phosphorylation, this very last step, is going to create somewhere around 26 to 28 ATP. And we'll talk about why there's a range coming up here soon. So from cellular respiration, we yield a total, so the grand total, including what we create from glycolysis and the citric acid cycle and oxidative phosphorylation, we yield a total of 30 to 32 ATP from a single 1 glucose molecule. So the range is due to the electron transport chain and where NADH and FADH2 enter it. So if you remember, I told you to put a little star next to that information when we were talking about it before. And that is because it causes this little bit of fluctuation where we create either a little more or a little less energy. Because when FADH2 enters that second protein in the chain, it is going to have less energy because it's not starting at that first protein where it would create more energy by going through all 4 of those protein molecules. It's starting at the second one and therefore not creating as much energy. And that's what causes that range in ATP in the end. So remember that oxidative phosphorylation occurs all along that inner mitochondrial membrane, so where we see all those ridges and curves in this illustration here in orange. So it's the lighter orange that we're looking at. That's that inner mitochondrial membrane. So those folds, which we know are called cristae, increase the surface area of the membrane. And the reason that we want to increase the surface area is so that we can have as many of these electron transport chains and ATP synthase embedded in that membrane as possible within the mitochondria. So this mass production of ATP happens thousands of times within one mitochondrion, and overall, that is why we call the mitochondria the powerhouse of the cell. To conclude cellular respiration, our goal is to harvest energy as ATP from fuel in the form of glucose. And this is important to all cells because ATP is necessary to perform nearly all cellular functions. If you need a way to review, a good way to do that is to write down each step of cellular respiration. And remember that there are the 3 big steps, a smaller half step, and 2 sub-steps that we just talked about. And underneath each of them, you should write down what it is, where it's happening, why it's happening, or the goal of that process, and then the products and where the products are going to be going. That should give you a good idea of how well you're understanding the material.